Herein we are going to see a topic called as translation or protein synthesis, which is actually one important event that takes place during the cell growth and differentiation. So what exactly this transcription translation is? It's actually the conversion of the DNA sequence, the information of mRNA into an amino acid sequence. So there is no actually functional relationship between a DNA and actually a completed protein, because both of them are different kinds of biomolecules, and at the same time the DNA and the protein is connected by one molecule that is known as the tRNA or transfer RNA, which we will see in subsequent slides. So basically, for making protein within a cell, these things are required. The first thing is the messenger RNA, which is in having all the information for the making the protein in the form of codons. Transfer RNA, which brings the appropriately charged amino acid to the site of protein synthesis, and a very powerful enzyme called as amino acid tRNA synthetase, which is actually supposed to bind the amino acid with the corresponding tRNA. And we have a molecule, actually it is actually a protein making machine called as a ribosome, in which the proteins are correctly manufactured as per the genetic code. And as we know, it has two subunits. Both the subunits are involved in the protein formation. So coming to the messenger RNA, this is the basis on which proteins are built, and it doesn't have any non-coding region as we see. So it is translated into directly the protein. So a sequence on the mRNA that has a start codon and is completed with stop codon is known as an open reading frame. An open reading frame will specify only one particular protein. And as we see, the direction of translation is from the 5' end to the 3' end, and all the nucleotides are read in 3 per turn, which is known as a codon. We have two important codons here. One is called as AUG, or the start codon. It has to specify the beginning of the amino acid polypeptide chain, and we have three stop codons, UAA, UAG and UGA, which will stop the translational process. And the difference is, in prokaryotic mRNA we have more than one same protein made by one particular mRNA, which is called as polycystonic mRNA, whereas eukaryotic mRNA have only one single ORF for protein synthesis. So the first event is actually the recognition of the mRNA by the ribosome binding site to the beginning of the translation. So in prokaryotes there is a sequence called the Shine-Dalgarno sequence, which will help the 16S ribosomal RNA present in the 30S subunit to correctly anchor the mRNA for translation, and the adenine and the uracil and guanine residues in the 16S mRNA, ribosomal RNA, will actually bind to that with the mRNA and secure the mRNA for translation. And this is the very important step in initiation of translation with regards to bacterial protein synthesis. In eukaryotes, it uses a different machinery, wherein we know that eukaryotic mRNA has a 5' cap. So the eukaryotic mRNA 5' cap will be first sought out, and then after the ribosome binds there, it will move downstream till it occurs start codon. This is called as scanning mode of eukaryotic translation. Apart from that, eukaryotes have two sequence elements. One sequence, the 5' end, more or less similar to the ribosome binding site, it's known as Kozak sequence, and a poly A tail is seen at the end of the mRNA, which will actually give more strength to the mRNA, and it will also help in again the translation process. So we are going with the third species, that is known as the transfer RNA, which will actually bring the correct amino acid to the corresponding site of the translation or the protein synthesis. So they have three loops like this. This is the anticodon loop, which will recognize the codon, and this is the 5', 3' acceptor end, which will fuse the incoming amino acid, and it will generate the amino acid tRNA. So the enzyme which powers this is called as amino acid tRNA synthase, and an tRNA which is without an amino acid is called as an uncharged tRNA. The biochemistry of this enzyme involves two steps. In first, the amino acid and the ATP are added together. It is activated to form adenylated amino acid, and polyphosphate is removed in this. This polyphosphate is actually triggering the process of creating the amino acid tRNA. In the second step, the activated amino acid will now bind to the 3' end of the tRNA and form an amino acyl tRNA. And isoaccepting tRNA means the types of tRNA molecule, various types of tRNA molecule carrying a specific amino acid. Always remember that charging of the tRNA is very accurate. Only one particular amino acid will bind to a corresponding tRNA, and if the incorrect amino acid comes there, the proofreading activity will ensure that the amino acyl tRNA is not manufactured. So we have the biochemistry of this. Finally, we are going to the ribosomes, which are going to manufacture the protein, and as expected, they are composed of two subunits. Particularly the eukaryotic ribosomes are given here, prokaryotic ribosomes are given here, and ribosomes have to be assembled and disassembled during a translation process. An mRNA having a lot of ribosome is called polyribosome. So here we are going to see the first event wherein translation initiation is taken place. So ribosomes have three binding sites. One is called the P site and the next one is called A site. So in this particular picture, 30th ribosome subunit is already activated and by initiation factors and GTP. So the mRNA is actually brought into the place, and the first tRNA that comes there is called formyl methionine tRNA. So once the binding has taken place, the mRNA is secured in its place. The A site is now free. So in the elongation step, what happens? The A site is occupied by the corresponding tRNA, and once both the sites are occupied, a peptide bond is formed by the peptidyl transferase. And finally, once the peptide bond is formed, the ribosome advances to the next codon, thereby releasing the corresponding tRNA, and the process will continue till the entire polypeptide is completed. So three stop codons are known as UAA, UAG and UGA. They actually terminate the process of protein synthesis.